Hi there. In this lecture, we're going to learn the anatomy of a phage particle and try to connect the different phage proteins that make up the phage particle to the genes that encode those proteins in a phage genome. So last semester, we learned the anatomy of all of the phages that belong to Virales, which are tailed phages. And we spent a lot of time talking about the anatomy of Siphoviridae phage particles, mainly because about 96% of phages fall into this category. And these are phages with an icosahedral head, which is a 20-faced shape with 12 vertices, 12 little points, and it has a long, flexible, non-contractile tail. But what are the different proteins that make up this capsid? So one of the first proteins that I want you to learn about is called the major capsid protein. And it's the major capsid protein because it's the protein that essentially makes up this icosahedral head. And there are other minor capsid proteins that contribute to this head, but this is the major subunit of this capsid. And what we're showing here is the major capsid protein from a rather famous phage called HK97. And this single protein makes up the majority of this capsid. So the next protein that I want you to learn about is the portal protein. And the portal protein forms a dodecameric ring, which essentially means 12 portal proteins come together to form a ring, and they kind of form the opening of the 12th vertex of this icosahedral capsid. So let's talk a little bit more about how the major capsid protein assembles to form this icosahedral head. And this has a lot of text on it so that you guys can have this text to look at later. But what I want you to do for this video, instead of reading the text, I want you to pay attention to this, this little figure down here, and you can read this text later. And also this text, this exact same text and slide, is found in the content of your OneNote notebooks. So you can access this text at another time. So the capsid proteins assemble to form this icosahedral head, and they do so by assembling around the portal protein, which is shown here. And again, this is a dodecameric ring of proteins that form the opening to the capsid. And there's a second protein involved in this process, and it's called the scaffolding protein. And the scaffolding protein does exactly what you think this protein might do. It helps facilitate the assembly of the capsid protein into this icosahedral head. And so once this happens, what we have is a pre-head or pro-head rather complex that has the major capsid protein, but within are all the scaffolding proteins that are sort of stabilizing the head until the terminase comes along and packages the DNA. And as the DNA is packaged, the head matures. And during that maturation process, there's another protein called the capsid maturation protease. And this protease means that it can cleave proteins. And that's what it does. It's going to make little clips and cleaves of proteins in the prohead that essentially remove the scaffolding proteins, allow the major capsid protein to take its final form, and allow the DNA to be packaged inside the head. The other thing I wanted to point out that's kind of nice in this figure, so the blue is showing the portal proteins. After the capsid is packaged with the DNA, there's some neck proteins that we call head-to-tail connector proteins. And there's usually 4 or 5 of these in a phage genome. I'm going to talk about these more on the next slide. But what these proteins do is they connect the capsid to the tail, because the tail is added on after the capsid is packaged with the DNA. And they do two things. One is they kind of act like a plug to keep the DNA from coming back out, and form a connection with the tail protein. So let's talk about these head to tail connector proteins next. So like I said, there's usually 4 or 5, and they're very small little genes. But they're in general called head to tail connector proteins, but we are only allowed to call 3 of these genes in a phage genome, and mainly because we know the structure of these 3 proteins and they're easy for us to identify. The other 2 head to tail connector proteins we think are probably head to tail connector proteins, but it's not always easy for us to identify them. So there's one called the head to tail adapter, a second called the head to tail stopper, and then the final protein is called the tail terminator. So let's take a look at that. So here's another figure showing packaging of the capsid. This one shows a little bit more nicely, though, the role of the portal protein, which is shown in blue here. This red protein, which is GP15 in the bacillus phage, SPO1. And it kind of interacts with the portal protein. And then there's this stopper protein, which essentially acts like a little plug for the capsid. And so again, these form a bridge between the capsid head and the tail, and these proteins obviously are interacting with the opening to the capsid, so they're involved both in packaging, but they're also going to play a role when this phage goes and infect the next cell, and the DNA has to be released and go through the tail tube to get inside the cell. So the minor tail proteins is sort of a catch-all gene function that we assign to proteins that might be involved in these like tail fibers here, or the proteins that make up this base plate. So the tail fibers are what interact with the surface of the cell. So minor tail proteins that are involved in these tail fibers often are involved in interacting with sugars that are located on the surface of the host cell. So there's lots of sugar binding domains on these. And then some of the tail proteins, for example, those that are part of the base plate, are actually involved in penetrating the cell surface. So some of these might even have lysozyme activity. And reminder, lysozyme is an enzyme that cleaves peptidoglycan polymers. So the tape measure protein is a really, really long protein, and it forms a trimer. And a trimer is essentially 3 proteins in a complex. So 3 tape measure proteins kind of twist together to form a trimer. And it runs the length of the tail, and in fact, it determines the length of the phage tail. And so the longer the phage tail, the longer the tape measure protein gene is. And in fact, the tape measure protein gene is often the longest protein in the phage genome. It's not always true, but for most phages, this is true. It is also involved in injection of the phage DNA into the host cell, and sometimes it even has transmembrane domains in the protein because it's involved in interacting with the host cell membrane. Now, the major tail proteins are called the major tail protein because it's the protein that forms the major component of the tail. And these proteins essentially assemble around the tape measure protein and form what is called the tail tube. Now, the major tail proteins don't do this really well by themselves. They need some helper proteins called tail assembly chaperone proteins that help facilitate, one, the folding of these proteins, and then the assembly of the major tail proteins around the tape measure protein until there is a full tail formed. So we can find all of these genes on a phage genome. And in the other lecture, you learned about something called synteny, where the order that these genes are actually found on a phage genome are usually the same across Siphoviridae phages. And so you can look this up in your OneNote content tab. You can also look in the bioinformatic guide. But usually you have a small subunit terminase followed by a terminase subunit. We're going to talk about those soon. Followed by a portal protein. Then comes our capsid maturation protease, the major capsid protein. Here are these 5 head-to-tail connector proteins. And again, we can only call 3 out of the 5: head-to-tail adapter, head-to-tail stopper, tail terminator. Then comes the major tail protein. The tail assembly chaperone proteins are here. These are these proteins, or this gene that has the program ribosomal frameshift that we learned about last semester. That's followed by the tape measure protein. These guys are all the minor tail proteins. And then way down here on the right in this box are genes that are involved in lysis, and therefore we call this the lysis cassette. And a cassette is essentially a group of genes that are adjacent to each other on a chromosome or a genome that are involved or play a role in the same process. So let's talk about the lysis cassette. There are 3 genes that we will find in mycobacteria phages. And there's a little caveat here for those of you studying curtobacteria phages. As you know, mycobacterium has 3 layers of the cell. It has a cell membrane, then it has a peptidoglycan, followed by the outer membrane, which is made up of mycolic acids. And that's shown right here: plasma membrane, peptidoglycan, and then that mycolic acid outer membrane. And so phages that have been assembled inside a cell that's infected have to get through those 3 layers in order to go on and find new host cells to infect. And so they encode 3 different genes that cause lysis of the cell. I'm going to talk about this one on the top first. This is lysin. I'm going to talk about holin first. Holin, we can recognize holin in a genome. It usually has 2 transmembrane domains, which are parts of the protein that love to be stuffed into the hydrophobic membrane. And this allows, here's like one green protein shown here. They kind of assemble into a pore, so multiple holin proteins come together to form a pore. And that pore allows 2 other phage-encoded proteins called lysin A and lysin B to pass through the pore and then have access to the peptidoglycan and the outer membrane. The first one is called lysin A, and it is essentially an endolysin. And what it does is it cleaves the peptidoglycan layer. It degrades the peptidoglycan layer. This allows lysin B to have access to this waxy mycolic acid outer membrane, and it cleaves bonds between the arabinogalactin sugars here and the mycolic acid fatty acids. And that in its is what ruptures the cell and allows these phages to escape the cell. You can find these 3 genes usually after the minor tail proteins. And I think what you'll find is across the different phages, most of the time we find this cassette after the minor tail protein. But in some phases, like for example, the cluster A's, they can be located earlier in the genome. Finally, I want to talk about the terminase genes. Clearly, I'm making this lecture with my dog. Hey, sit down. We'll let him scamper off and... Okay, the terminase genes. There's usually two terminase genes found in a phage genome. One is called the small subunit terminase, and its job is to bind DNA, and it has a DNA binding domain, and it binds specifically to sequences in the phage genome. This allows the terminase to interact with phage DNA rather than host DNA. The large terminase works with the small terminase, and it has two different functions. It has one domain that has nuclease activity, which means it can cut DNA, and it has another domain that has ATPase activity. This domain acts as a molecular motor. And what this does is allows the terminase to translocate the phage genome into the capsid head and make cuts at appropriate points in the phage genome so that there's a single phage genome unit inside the capsid head. So let's look at what this looks like. Here's a phage terminase. It has recognized a sequence in this long concatemer of DNA, which you guys learned last semester, is that when phage DNA replicates, the result is actually usually a really, really long piece of DNA that has many, many copies of phage genome units in it. It will interact with the portal proteins and begin to translocate the DNA into the capsid, and it uses ATP in order to do that. When the entire genome is inside the cell, usually it recognizes another sequence and makes a cleavage there, such that one phage genome unit is inside the capsid.